Me back over here to talk with you today about literature searches. Can you all see my screen?
Yes.
All right, great. All right, so hi, I'm Jessie. As Jenny said, I'm currently a senior clinical scientist at GeneDx, where I spend most of my time these days doing exome analysis work. But I've been involved with ClinGen since 2014. I'm currently co-chairing the P10 VSEP and member of all the other groups Jenny noted. And I'm working remotely from the Cleveland, Ohio area. Prior to my work as an exome analyst here at GeneDx, I was mostly involved with the hereditary cancer team doing variant interpretation and report writing, which is where I have my major background with respect to doing literature searches for variant workups. And so you might recognize my name from this lecture, which you I think is required for you guys to do when you're doing your Biocurator training on the ClinGen website, recorded back in 2019. So for our time together, I'm not going to rehash everything that's in that video that you've already watched, but we are going to re-review some of the basics, and I'm going to add in some, I guess, some time-saving suggestions that have, that for me are what kind of my go-tos for when I'm reviewing a variant and trying to be as efficient as possible. And we'll also be addressing all of the questions that all of you sent in prior to the talk today. So thank you in advance to all of you who sent in questions ahead of time. I do want to say that this presentation is going to focus on variant vetting. Gene vetting literature review is also a great topic, but is definitely out of my scope of expertise. So I've suggested that we do a separate presentation on that topic at a later date. Okay, so first off, why do we do a literature search? We're trying to find articles that allow us to apply ACMG criteria during variant curation. So we're looking for functional studies. We're looking for our case reports, especially ones that help us understand if a variant occurred de novo or we have really solid phenotype information. We also might be looking for studies that characterize the mechanism of the variant as well as case control data. And the evidence we find might be for or against pathogenicity. I will say that clinicians also perform literature searches as well, possibly more focused on the gene than on the variant to help them understand the phenotype spectrum and, excuse me, and whether this variant, is a solid fit for the patient that they have in front of them. But when they're performing a literature search, they might be looking for additional information on a variant that might not be included in a laboratory's report. So from a laboratory perspective, it's why we always try to be very thorough and capture all of the information possible to save our clinician friends' work. So some shortcuts to finding helpful information. So my first go-to these days is to look at ClinVar. So this is not cheating, okay? This is not, a contest to see how good you are at searching Google to finding literature. Okay, you're trying to find information that helps you classify the variant as best as possible and as efficiently as possible. So it is completely fine to look at ClinVar and see, who has already put information out there on this variant, and cited sources that you can refer to help you in your work. So we're going to be looking at a P10 example because why not? And so this P10 variant, arginine 15 serine, pathogenic, likely pathogenic per a couple submitters. And we click on the more link over here for both of them. We get the full evidence details, which for both of these summaries nicely describes the information and cites all of their sources, either by PubMed ID or per the author year information. So you can look it up and find the information to perhaps apply to your case as well. And so all those literatures that are cited are all also at the bottom of the page of the variant page in ClinVar with their PubMed ID links to make it easy to find the information and review for yourself. So that is a point I do want to make is, this information, it might be so nicely described in the summaries. We probably didn't mess up and make any errors, but it's still very important to look at the cited articles yourself. So Maybe a submitter might have cited a paper, but it doesn't actually study the variant you are interested in. Maybe it studied another missense change at the same residue. And so the paper is cited in ClinVar for that purpose, to implicate the importance of the residue. But you want to look at it to make sure that the variant that you are interested in is actually the one that's studied. Some of the publications, they might say, oh, it's, patients that, all have such and such phenotype, or they're not closely described in the ClinVar summary, but you want to look at the article yourself to see if maybe they do have a supplemental table that gives detailed clinical information about the patient that is useful to you. Or maybe the opposite is true. Maybe it's just a large cohort study with really zero information, and it doesn't include the information of the detail level that your VSEP requires for applying criteria. So you just have to look at what's in the paper yourself, look at any supplemental data to see really if you're able to apply any criteria. Another thing is that authors and reviewers for articles they're human. Sometimes papers do have small nomenclature errors that give you less confidence that your variant is actually the variant that's in the paper. So another excellent reason to definitely look at the paper yourself and really closely look at the nomenclature to make sure that it is your variant that's published. And again, although in a ClinVar summary, the submitter might say, oh, we applied PS2 because this person was de novo, but maybe the paper doesn't actually state that that parental or paternity and maternity were confirmed. If it was a West trio study, okay, well then you have good indication of that. But some of them just do a singleton analysis and then do targeted follow-up later. So PS2 actually isn't appropriate. So it's good to, again, review the paper to make sure that what the criteria that are being applied by the submitter matches with how your VSEP would also apply criteria. Another resource that I'm sure all of you know and love by this point is HGMD. So HGMD, as you know, lists variants found in individuals with a particular disease. I'm so sorry, I'm getting a little bit of a cold, so I'm going to take a sip of water real quick. All right. In HGMD, great resource, has a free version. What a downfall is that it's not very up to date, but for a free resource, you really can't complain. It does provide citations and links for all of the papers that are included. However, sometimes those links take you to the PubMed homepage rather than the listing for the publication itself. So just beware. And like I noted, there is a free version that's available. Registration is still required, and it's less updated than the paid version and not as pretty to look at. HGMD also provides classifications on the variants that they list, but big red flag over here, like do not trust this with a grain of salt, okay? So their abbreviations are, they use DM for disease causing mutation, DM question mark for likely disease causing, and some other things for disease associated polymorphisms, or something that might just have some laboratory data that might be supporting a functional impact. I have no idea who makes these classifications. They're not classified according to ACMG criteria. So again, look at the papers on your own, think critically, and apply, or maybe don't apply, evidence per your regular VSEP processes, depending on what information is present. HGMD is also something that people look to when they're trying to figure out if they might apply PM1, so our hotspot code, other missense variants at the same residue are pathogenic, or I'm sorry, missense variants around the position are pathogenic, or PM5 missense variants at that same position are pathogenic. And so you might look up in HGMD and say, oh, there's several variants that are at the same or the nearby residues that are DMs. They're called disease-causing HGMD. Can I just say, okay, this is great, and use these variants to apply PM1 or PM5 per my VSEP rules just based on this info alone? No, the answer is no, don't do this. Go for that variant. HGMD is a great resource to learn about the existence of those published variants at the same or nearby residues, but you still need to work them up per your standard VSEP curation processes. And then if those variants meet pathogenic or likely pathogenic all on their own, then you can use them for, PM5, PM1, whatever code you are interested in. So we're going to take an example look at HGMD again using P10 over here. So this is the free version of HGMD. Here's our gene, here's P10. And then on the table over here, it lists all the different variant types. We have missense and nonsense grouped together, splicing, regulatory, et cetera, and the number of variants that they have listed in those categories. So when you click over here on the Get Mutations button, it lists out in codon numbering order all the variants that they have listed in the free version of the database over here, as well as with citations for the publications. Again, most of these do not actually take you to the PubMed link. So again, I think HGMD is a really helpful resource to find nearby variants to work up for consideration of PM1 and PM5, in addition to literature, of course, on the variant that you are interested in. Finding nearby variants is also possible to do in ClinVar. For me personally, I have not found a more efficient way to do this. If you know of one, please speak up. But by searching ClinVar, if you limit it to molecular consequence of missense and sort by location, which is the default, you can find your residue of interest and see what's nearby. So let's say if we're interested in the ARG15 residue, maybe we found, ARG15 threonine, and we want to understand, okay, what if there are other variants that are in the neighborhood that are also classified, or we might have helpful information on to potentially classify. We can say, oh yeah, there are some nearby variants, like this one has been classified likely pathogenic by the expert panel. That's great. As well as some variants at ARG15 serine as well that are pathogenic or likely pathogenic per multiple submitters. So these, again, would be ones to be able to look at further to consider whether you can apply PM5 criteria to your variant of interest. I think another thing that we often are challenged by is to understand how much lit searching is enough. So how deep do you dig? How far do you look for information? So I have a couple of situations over here that we're going to talk through. There might not be a perfect answer for any of these, but I'm just going to share my perspective on kind of when I call it quits on looking for information. So the first scenario I'm going to speak to is, let's say we're vetting a loss of function variant for a rare autosomal dominant disorder for which loss of function is the disease mechanism. Okay, great. So we have a loss of function variant in a gene where loss of function is the mechanism. We get to apply PAVS1. We are well on our way to getting to pathogenic or likely pathogenic. We see the variant is absent in NOMAD. Okay, fantastic. We get to apply, our PM2 probably at the supporting level code these days. Based on this information alone, you're already at likely pathogenic. Great. Maybe per your VSEP rules, you just need to find one patient with a good phenotype to get you all the way to pathogenic. You look up your variant in ClinVar. There's an entry that has a great case report that gets you PS4 supporting or whatever code your VSEP lets you use, PP4 maybe. You are now classifying your variant as pathogenic. Are you done? Are you okay to stop there? My answer is maybe it might be okay to stop there. Some things that I would think about is if the publication cited in ClinVar is from several years ago, I might still do a little digging to look at other resources, maybe do a little quickie Google Scholar search to see if there's a newer report to add in with additional information. Maybe there's, de novo data on the case, if that makes sense for that gene. So if it's from several years ago, yeah, I might look around and just see if there's anything new out there. But if the publication that's cited in ClinVar is very recent, like maybe it's only a year or so, or newer, it's probably unlikely that you're going to find any new information if you do more extensive searching, like in Google or Google Scholar. The submitter likely would have found and included additional older relevant case reports in their summary. Again, I don't think it hurts to quickly check HGMD to see if there's anything older that's listed, but I'm saying that if you're playing the odds over here, it's very unlikely that you're going to find new information. And if you do find new information, again, based on the evidence you already have to date, a rare, absent variant in Nomad that's loss of function in the gene where all of this makes sense, it's unlikely you're going to find any new information that makes you walk that classification back anyways. But this is just my perspective on this. If your VSEP leadership or senior biocurators tell you to practice differently, just do what they say. But I'm saying don't kill yourself to find more literature when you already have enough and it's pretty new information that gets you all the way to pathogenic. Here's another scenario. All right, you're working up a novel missense change in a gene where missenses can be disease-causing. And your patient is de novo via an exome trio and is a nice fit for the phenotype. So let's say you get to apply PS2 as well as PS4 supporting. The variant's absent in Nomad. It's at a hotspot residue where you've seen pathogenic missenses previously. We're starting to rack up our criteria over here. There's a recent case report noted in the ClinVar summaries that also fits the phenotype, but they don't give you a lot of specifics, no more criteria there. In silico predicts it's pathogenic, which agrees with everything else you found. So based on everything you found so far, you've got a ton of criteria, including that strong PS2. You're already at pathogenic. There are 2 ClinVar submitters that are also at pathogenic. Nobody is at uncertain or any other classification, so there doesn't seem to be anything yet that's conflicting. Question here is it worth it to go digging for additional evidence? For this one, I actually would. So for this missense variant, so far you don't have any functional evidence that supports this variant being damaging. This information might not exist, but if it does, it would be good to review and be able to include in the ClinVar summary. Maybe the missense variant has had a functional study that contradicts pathogenicity. I mean, probably not, given all the other info we have, but good to be thorough. If it supports PS3, it nails down your path classification, and then you're really done. But if it's contradictory, it could push the variant back to VUS. Another thing to keep in mind is that de novo variation happens in the general population, and they can be red herrings. So I guess when you see it, it does give you confidence. It gives you great criteria. But I guess I would feel better calling myself done with a missense variant if I also had a functional study. So I would go looking for that bit of information. But again, if I didn't find it, then that's okay too. All right, let's go scenario number 3. We have a synonymous variant that has no splice impact predicted. So we have BP4 and BP7. The variant also has a lot of population data, and it meets your VSEP's BS1 criteria. So based on all of this information, you are at a classification of likely benign. The question here is, do you need to do a literature search to find more information about this variant? My feeling on this issue, again with the same disclaimer as before, is probably not. Because what are you going to find? You're going to find some information that might have documented this variant in a patient with some, maybe some phenotype that fits the disorder. But VSEPs are advised by the SVI to not apply proband counting or segregation criteria to variants that meet the BS1 threshold. And the reason for that is because these are more than likely coincidental observations, and it's going to lead to you racking up proband counts and myoses just due to the high population frequency of the variant. My suggestion for when you encounter this scenario is to take a look at ClinVar and see if any submitters have classified the variant as anything besides benign or likely benign, and review what evidence might have been provided. Sometimes in this situation, you'll come out with an OMIM entry that might classify it as pathogenic. But again, some of these are quite old. They're before we had the advantage of population data to help us understand population frequency of some variants. So it could very well be a coincidental case report. but still, maybe skim the article for what information was provided. It's also possible the OMIM entry has functional data that might also support you applying BS3 and getting you all the way to benign, and then you are done with the variant. Checking HGMD for the variant might also provide you some similar information that again gets you all the way to benign, meaning that you don't have to revet the variant again per the ClinVar, or I guess ClinGen variant workup processes. If a submitter is at VUS, if it was from several years ago, they might not have had the advantage of current NOMAD data. So I would take a look and see if they had any evidence provided to support their VUS assertion instead of benign or likely benign, and check it out. But if you don't see anything at first glance that makes you really question your likely benign classification, I might be okay stopping there. Again, you could search a little bit to try to find more information that really puts the nail in the coffin and puts you all the way to benign. But I think you're also okay to not spend hours and hours looking for more information on the variant. Next, I was going to talk a little bit about variant revets, what type of information to look for. So when I say the word revet, that might be a little bit of lab speak. What I'm meaning is recurating a variant to look for new evidence to support if you can change your classification. So per the processes we use for our ClinGen VSEPs, we're supposed to be doing this for likely pathogenic or variants of uncertain significance every 2 years. For likely benign variants, when a new NOMAD release is available that might give you POP data that puts you all the way to benign. But if you get a variant all the way to pathogenic or all the way to benign, like, you do not have to revet the variant. So it's one of the benefits of being able to find information to get you, either all the way there on either direction. So if a variant isn't likely pathogenic or uncertain, I think the most critical things to look for is what has changed since the last time the variant was reviewed. Is there a new submitter in ClinVar? Bug them, poke them, see if they have internal data that might be able to add to your criteria and help you reach a different classification. Sometimes a ClinVar submitter might have updated their classification. Maybe they went from likely pathogenic all the way to pathogenic. This is another reason to reach out to them and say, like, hey, what did you find? Did you find a new, like a homozygous case that fit the phenotype or something? If it's a recessive disorder that helped you update your classification. I think it's a great idea to reach out to ClinVar submitters for new information like that can help you perhaps find new information to change your classification. It's also okay to limit your search to new literature to the time point of the last review. So for example, let's say you had a variant that was reviewed by your VSEP in March of 2020. Your focus should be on new information from 2020 to present. I would feel confident, everybody that I've worked with as a curator has been, great and detailed. I would trust that they did what they were supposed to do and found and reviewed new literature prior to that 2020 time point. So I would focus on things that are new from there. In HGMD, the professional version, I don't think this is in the free version, but the professional version, if you have access to, provides the date the paper was included in HGMD. And sometimes that doesn't totally align with the date of the publication. So this is an ATM variant that's published in Jackson 2016. If let's say you're reviewing the variant, again, our situation, we last reviewed it in 2020. You might think, oh, well, this paper is from 2016. Obviously, it was taken into consideration when we thought about the variant previously. And maybe maybe they forgot to add it to the VCI, or maybe there weren't criteria that were applied when they reviewed it, so they didn't need to add it to the VCI. But I'm just going to ignore it. I would say don't really do that. When you look at the date the variant was added to ClinVar, it was only included in September of 2021. So although the paper is from 2016, whatever process happens to get papers added to HGMD did not happen until 2021. It wasn't discovered and added until then. So this would be a new paper, even though it's dated 2016, that might not have been easily found and taken into consideration when the review in 2020 was performed. Bottom line, HGMD doesn't have that many publications on each variant, and it's also a pretty easy resource to quickly skim. So I would just recommend just take a look at HGMD to see if anything is newly added or wasn't maybe included in the VCI when the variant was first reviewed, to see if it was taken into consideration. You can also set date limits on Google searches. So in Scholar, there's this little bar on the left-hand side where, if you just want to do new stuff since some year point, you can just click there and it'll get you there. Initially, so I just did an example TP53 variant. Initially got 19 results for it in Google Scholar, but when I added a time frame of just doing 2020 onwards, it reduced me down to only 10 papers to kind of skim and review. So again, 19 is not terrible. It's not thousands of papers, but let's be efficient where we can. I think it's fine to limit your search range to the last time point that the variant was vetted. And Google, the the impact is even more significant just because Google just gets you so much mess sometimes. But initially you get 799 results for this variant. If you go to the tools thing up here, it gets you this little drop down where you can say add a time frame. And so we're just going to do custom date range from 2020 to just leave that blank. It'll take you to present. And now, again, we only have 10 results. So that's way better than 799. And just so you know, you don't get this number right away, but I just hit tools again and it gets the number of new search hits to show up there. All right, so those were all the things that I was thinking would be great to cover, but then all of you came up with some other questions that we also wanted to take a lot of time to talk through as well. Somebody had a question of how to better search for specific intron variants. Great question. So I think that incorporating the IVS number into the nomenclature search is helpful. So what is this? So this is not HGMD nomenclature. IVS, it just means intervening sequence for intronic variants, and it has to do with the exon numbering. So for this ATM variant that we found that 2016 paper before. This is just another way that sometimes publications will use to describe variant nomenclature. They might not use CDOT. They might just use, say, what intron we're dealing with over here. So ATM CDOT 2639 minus 22 is an exon. It's an intron 17. It's a 3 nucleotide deletion. It might be published instead because it's an intron 17 as IVS 17 minus 22. IVS 17 minus 20 del. When you're doing this, when you're using IVS, it's good to be aware of possible changes to exon numbering for your gene over time. So for ATM, this is particularly relevant because once upon a time, ATM was believed to have 65 coding exons. The current transcript that's used for reporting has 63, but old literature will list this variant as IVS 19 instead of IVS 17 based on that old nomenclature information. So when you're first starting out, especially getting to know a gene, you might go, oh my God, how do I even know if this is a thing? How do I know if this is an issue for the gene that I'm trying to search, trying to be thorough and find all the literature on? This kind of information sometimes just comes with knowing your gene. Expert panel members are likely aware if this is an issue because some of them have probably been, looking into this gene and familiar with it for decades. So I think that it's helpful for the expert panel to share with their biocuration team if there is alternate nomenclature that they need to be aware of. Maybe there was a different CDOT position that used to be used prior to prior to currently HGVS standards. Also, when you find other papers, when you're searching for literature, this can sometimes provide you a helpful clue that you have this situation to deal with. So, all right, so we're going to try to look up this ATM variant. Again, that's in an intron 17. Looking it up in ClinVar, it's pathogenic, likely pathogenic for some submitters. That's cool. And then the citations for the variant, you have several publications over here. Okay, wonderful. Let's look at some of them and see what we find. So for this Nakamura paper over here, you find your variant by the CDOT nomenclature, but it's called IVS 19. And it says, this variant is in patient whoever, creates a novel splice acceptor site in intron 19. At first, when you find this, you might go, well, if you're not aware of the alternate exon numbering issue, you might go, maybe they just made a mistake. Maybe this isn't not really how, maybe they just messed up and said 19 instead of 17. But when you look further at other information in the paper, they do the same thing. For this other variant that's at the same position in the same intronic sequence, we're also saying intron 19 instead of intron 17. This would, for me, kind of put the nail in the coffin that we're dealing with alternate exon numbering that you need to be aware of. So based on this, if I'm in a situation where I really need to dig for more information on the variant, in addition to IVS 17 being a part of my search string in the resources that we look to, I would also include IVS 19 as well. Does this result in a lengthy search string? Yes. But it's one search string, and hopefully will help you find all the information to be as thorough as possible. And another thing is, if you look at this variant in HGMD, again, might be hard to find because you're thinking this is an intron 17, not 19. But if you're clued into the kind of unique nature of the intronic position being minus 384 eta G, you're also saying that this Nakamura paper overlaps with the ClinVar citation. All signs are pointing to this being the same variant as well. Another good question that somebody sent in ahead of time is how to better search CNVs with specific or overlapping breakpoints. I will just say in general, CNV searching is not easy. It is not as straightforward as searching for sequence variants. They can be difficult to find in ClinVar due to differing breakpoints, sometimes the builds that are used, and just having to kind of understand the position of the breakpoints for your CNV of interest. Internally, for my daily life, we have found HGMD to be the quickest and easiest way to find CNVs that match up with the event you are interested in. All right, so going back again to the free version of HGMD, now we're in the ATM gene. We have some options over here to help us find gross deletions, insertions or duplications, or complex rearrangements. So again, clicking on, let's say we're interested in a deletion, click on that. You kind of get a hot mess. So it's not very well organized. The paid version of HGMD, I will say, is much nicer and more easily organized and makes it a lot easier to find a specific exon deletion that you might be interested in. But really for this, you have to have some information maybe about the codon or nucleotide position to see if one of these 73 deletions might align with the one you are interested in. Some of them we do have exon numbering over here, but again, some of them don't. Just we're not making our lives easy, are we? Doing searches in Google and Scholar, I find that especially for single exon events, this is really hard because you get a lot of red herrings. It often identifies a paper that talks about, oh, there was a point mutation in exon 5, or maybe a deletion of a single nucleotide in exon 5, and it doesn't. It's kind of hard to narrow down. We've got 49,000 results over here. Like, no, thank you. To what is actually a deletion of an exon. It's really tricky. Multi-exon events are a little bit easier because you can use some quotes over here and include the full, exons 16 to 18 that you're interested in. Doing this, we have, fewer searches, but some papers are also going to give you methodology that has something special to say about those exons that, again, might not be a deletion was detected. But we can see here, this is actually the ClinVar entry for this event. It helps you find the ClinVar entry. It says this is a deletion encompassing exon 16 to 18 of BRCA1. Okay, wonderful. I would look at that. So a suggestion for specific breakpoints is welcome. If you have an event that has defined breakpoints, my first intuition would be to incorporate those coordinates into your search string, as well as if if the breakpoints include an exon or several exons, include that as well, just in case the event is kind of close enough to allow you to apply criteria per your VSEP rules. So for example, let's say we have a deletion in NSD1 causes Soto syndrome. And we know our breakpoints. At least we know one of our breakpoints, let's say, and it encompasses exon 13. Maybe we don't know the other breakpoint. I would include both deletion exon 13 as well as the coordinate for the breakpoint, just to see what you get, to see if there's anything helpful that pops. Another question that we had submitted is, how trusting can we be of information in LOVD? So LOVD, you guys are all probably already all familiar with it, the Leiden open variant, I think, variation database. And it can also be a really neat source of internal laboratory data, especially from maybe, I think some of the clinical submitters are from Europe, from European clinical groups, that they do put helpful data into LOVD. Sometimes the entry states the variant was de novo. Sometimes it's where you can get phase information, and it's not in any publication. It's just patients from a clinical setting, and it's unpublished from the submitter. But if that's all you find, if let's say you're looking for more info and you don't find anything else on this variant except for this LOVD entry, would you use this? Would you just rely on this to apply criteria? I will say that in general, I have found this information to be trustworthy. And in my daily working life, we have used it before to apply criteria, in particular when other information is already supporting pathogenicity, and the LOVD entry is saying de novo. You might think, oh, isn't that a little unfair to use it in some situations and not in others? Yeah, but what we're trying to do is, do the best job we can for the patient. And if, we already have one de novo observation in-house, we have this second one in LOVD, we're confident they're two separate individuals. I wouldn't want to let that hold me back from applying stronger criteria and perhaps providing a better classification for the patient. My suggestion would be, when you counter this situation and there's a submitter in LOVD that looks to have helpful information, reach out to the submitter. I've had good luck with this before. I found the folks that I've reached out to through LOVD to be great at quickly responding and providing helpful clinical details too. This is also your opportunity. Let's say the LOVD entry just says it's de novo. Maybe you would like some, more phenotype details that might also support, your PP4 or PS4 code. This is your opportunity to maybe get that information. So give it a shot. What's it going to hurt? But if they don't respond to you, and you don't get lucky, I would suggest reviewing this information with the full VSEP. Maybe the VSEP still feels confident applying criteria based on the LOVD entry. Maybe they do not. They might be a little bit more conservative if this is the one piece of information that pushes you from VUS to likely pathogenic. But I think this is a situation that requires full discussion, but still great thing to consider. So this is just a sample LOVD entry for an NF1 variant. LOVD, we don't have a ton of detailed information, but it does say origin de novo here. Okay, that's cool. So if we click on the individual ID, it takes us to this other page that, you get this huge scroll bar, you scroll all the way across and you get the owner. So when you hover over the owner name is when you get the email address for the person who submitted to LOVD. And that's who I would reach out to try to confirm the de novo information and see if you can get more clinical details. We also had a question submitted about somatic variant vetting, how to find if a gene or particular variant is a driver of somatic disease, like a given cancer subtype. So just so I'm being honest over here, my experience is very limited to germline variant interpretation. I'd welcome thoughts from others on who have somatic expertise. The first couple, but my background is in cancer, so I know I know a few things. I first thought of the Cosmic database. That's a place that I would go to look at if a variant might be a hotspot variant. This database is insane. It has a wealth of information. It lets you search by genes. It lets you narrow down to variants or residues of interest. Again, looking at Cosmic and P10, here it has thousands of variants and thousands of samples that have been identified to have P10 variants through some type of tumor testing. And it has the filter bar on the left-hand side where you can narrow down. Let's say if you're interested in, a specific missense position, you can narrow it down by amino acid. And it'll show you over here what are the different missense variants that have been reported in tumors before at those positions of interest. It'll also narrow down the tissue distribution. So, what types of tumors have these variants of interest been seen in most commonly, as well as a table that has, all of the variants, again, tissue distribution for any particular variant of interest. So this variant R130G is, very commonly identified in endometrial tumors, and as well as a lot of good literature that has PubMed ID links. I find this interesting, the confirmed somatic. I kind of wonder how they go about calling it confirmed somatic. But I guess that's a question for somebody who knows this database better than I do. My issue with the confirmed somatic thing is I actually kind of tested Cosmic by looking up a P10 variant that we've classified as likely benign. It has high population data. When you're doing tumor testing, you're not leaving out the germline. You are, you're testing the patient's tumor environment. They might have loss of heterozygosity of the wild type allele. They might not, and if they don't, they're going to have, you're going to be able to detect germline variants in tumor testing. So they call it, several of these confirmed somatic. I don't know if that was decided by allele fraction, if they're looking like mosaic variants. Well, that's certainly suggestive that it's tumor specific. I looked at some of the publications that I had access to, but I really didn't see information about allele fraction or other info that would confirm, one way or the other. So I think this is just something to maybe approach a little cautiously. The second thought I've had about finding somatic information, especially for maybe tumor hotspots, is to look at cancerhotspots.org, a resource produced by Memorial Sloan Kettering that finds variants in nearly 25,000 tumor samples. It also has a nice search function where you can search your gene of interest, and it comes up. You can order it by number of samples with variants and what specific variants are present at that residue. I think something to be cognizant of is if you're looking at this to maybe identify a hotspot for applying the PM1 code for a missense variant, you don't want to consider the information for a nonsense variant the same way. So for example, this R130 over here residue certainly looks like a missense hotspot. We have, over 100 different missense variants at that position, but the ARG233 right below it, the variants that are identified are exclusively a nonsense variant. So I wouldn't consider this as being useful for the PM1 code because again, it's a nonsense, it's not a missense at the same position. Okay. So I know that was kind of a marathon of you guys listening to me kind of blather about the things that I've learned and have helped me be more efficient. Oh gosh, I'm saying there's a bunch of questions in the chat over here. Oh no. But I wanted to open it up and let everybody chime in with any other input they had, or if I messed up or got anything wrong, please let me know. Or if you have more questions that you wanted to say to the groups. Okay, there's a comment over here from Saida. What is it meant by racking up proband counts of meiosis? I have not heard like this before. I am apologizing because I'm from the Midwest. When we say racking up, it means we are accumulating. So we are gathering up a high number of individuals with maybe a phenotype of interest just because we have identified the variant at a high population frequency and its coincidence. Let's see. Here's a question. What sources would you use for application of the PM5 criterion? Articles only, ClinVar, other databases? I hope I answered that question, that you do need to vet the variant yourself, but I think all of those areas are good ways. And I think in the main literature review presentation that's part of the curator training, I also talk about including the residue nomenclature in a search string to help you find literature on a residue using, Google or Scholar searches as well. So I hope we've addressed that, but if not, let me know. All right. Let's see. All right, so here's a question. ClinGen has set strict recommendations for consideration of functional evidence, e.g. for PS3. How do you deal with several articles and literature providing functional evidence for a variant, though not necessarily as recommended by ClinGen? Number of replicates, number of controls, etc. Great question. Probably I don't think that's the that's the main focus of our talk. But I will say, as somebody who is also on a VSEP and deals with that same situation, I think that the expert panel still has some professional judgment that they get to exercise. Yeah, we have the strict definitions for PS3. It's supporting at strong evidence level. You need to meet the threshold of evidence required for that. But I think that the VSEP also still has the ability to say, well, there are many functional studies that are showing an impact. Maybe we are not strictly meeting our VSEP rules, but we would be comfortable applying some lower evidence of PS3. I think the main point that I'm trying to say here is it might still be possible, but I think it requires discussion with the VSEP as a whole and reviewing the information together as a team to make a group decision. Yeah, and thanks, Dimitri, for pointing out the ClinGen guidelines on classifying somatic variants. That's definitely your area of expertise and not mine. All right. All right. Yeah, you're welcome, Konstantinos. Oh, so Tam, thank you for looking up. Confirmed somatic, the variant allele from the tumor sample differs from the germline alleles of the same individual. Okay. So maybe it's not just included in the source publication, not sure. All right, but I wanted to give you guys, I know we have about 13 more minutes here. Anybody have any other questions? You're free to go off mute if you want, I don't mind.
Jessie, I just want to say thanks so much for going through all of this. I think it's just really helpful to think about how to think about literature for specific codes as well. So this has been really helpful. Thank you.
Good. Well, while you guys think about if you want any other questions for discussion, I just wanted to say thank you again for inviting me to give the talk. Here's my email address if you want to reach out with questions following up, and some cute pictures of my cat.
So cute. Any other questions for Jessie? So Jessie, I guess one other question. So it sounds like the major sources you're using are ClinVar and Google Scholar. Those are the main sources for your literature searches. Are there other tools that you use, like LitVar or Mastermind, the free version, anything like that you've used, and what are your thoughts on those?
I'm actually I don't use Mastermind or LitVar. I found that my methods of searching are what I'm just comfortable with, because I guess it's what I've gotten efficient at using. I don't use whatever is helpful to you. But yeah, those when I kind of did a head-to-head comparison of my technique versus those other sources, mine gave me a better result.
Okay, that's great to know. Thanks. Okay, any other questions? All right, well, thank you so much, Jessie. This was just super helpful. I really appreciate it. Oh, there's one more question from Ben. Does anyone know how often Mastermind misses variants? I don't have a good answer to that. I don't know if anyone else who's used Mastermind has experience with this.
Yeah, I was just asking because I feel like, I don't know, I think they're pretty good. But
Yeah, I've used the free version. I use it pretty consistently, with the limitations, of course, that you're only allowed a certain number of searches, but I found it to be pretty good.
So we did a small study just for RYR1, and in our hands, we compared just looking at ClinVar or looking at Mastermind, or looking at HGMD, and Mastermind misses papers. Okay. So if you're trying to get everything, Mastermind often finds more obscure papers, but it doesn't always find all the papers that you might need. And I've asked them about it a couple times. And when I've like specific papers where I found them through other means, and they're always like, oh, well, that paper hasn't been annotated yet. And I was like, I thought it was like automatic. Like I thought they had like a web crawler or something, but yeah, so I know they do miss papers. So people should just.
Yeah, that's really good to know, Jennifer. And yeah, I thought the same. I thought they automatically download from PubMed like once a week or something like that. I don't know how regularly, but yeah, I was curious to know, is there something to do with the nomenclature? Is it missing older papers, things like that? But yeah, it's really, really good to know. I guess, yeah, no source is perfect. So.
But again, if you feel like you have mastered the use of Mastermind, and if that's a quick resource for you to use that might find that last paper that puts the nail in the coffin, yeah, great, go for it. I just didn't cover it here because it's not something that I'm used to, and I know that we do so many other talks about LitVar, and that's been covered before on other Biocurator talks.
Yeah, no, that's great, Jessie, and I think it's just really helpful to think about these, other methods as well, and having more than one method, like you're using, like you said, ClinVar and Google Scholar typically, but being able to kind of cross between the two, and where one might miss something and you'll pick it up another way. So I think that's just really helpful. Think about that. All right. Well, thank you so much, everybody. And thanks again, Jessie. So our next call is not until December 8th. So the next 3 calls are actually cancelled for various reasons. We have the ASHG meeting is at the same time as the 4th Thursday October call. So we're cancelling that one. And then it's a ClinGen meeting free week, and then it's Thanksgiving. So we will not be having this call again until December 8th, but I will be in touch with everyone before then to let you know about the topic, and look forward to talking with everybody then. And thanks again for joining today. And thank you, Jessie. This was really, really great.
You're welcome. Thanks, guys.
Take care. Bye, everybody.
Thank you.